Welcome to the video for chapter 11.3. So in 11.3, we're going to talk mostly about the protozoa category of eukaryotic microbes. So we're going to talk a little bit about amoebas, we're going to talk about some trypanosomes, some metamonads. So we're going to kind of hit some highlights of this group. So let's talk about section objectives real quick. So in 11.3, you need to come out of it being able to describe the form and motility of amoebas. And you need to be able to discuss the two diseases that we're going to talk about that are caused by amoebas. You need to be able to describe the distinctive traits of apicomplexan parasites. We need to be able to talk about Plasmodium falciparum and Plasmodium vivax, which are both eukaryotic parasites that cause malaria. We're going to talk about their life cycle. And then finally, you need to be able to talk about trypanosomes and metamonads like giardia. Let's first talk about amoebas. In general, amoebas can be divided into one of two groups. The amoebas that we normally think about when we hear the word amoeba is this first one, okay, which are called lobed amoebas. Okay, so they have these large pseudopods, these bulky pseudopods that they use to get around. Okay, this is the one that we normally think about when we think about amoebas. We have a second group, which is called the filamentous amoebas. Okay, so instead of having bulky pseudopods, they have these really thin needle-like pseudopods. Okay, so both types of amoebas are going to use these for motility. So they are going to extend the pseudopod out, okay, and kind of plant it like your foot, and then they'll drag the rest of the cell along with the pseudopod. They also use the pseudopods to capture prey, to eat, and feed themselves. Now we're going to briefly talk about a case history, which is called Spring Break Surprise. We have Brianna, who's a typical 20-year-old college student. She's in her senior year, so she wants to go on a really great spring break. So her and her friends go to Acapulco and have fun partying and just enjoying the beach atmosphere down there. Now, Brianna comes back and she brought something back from Mexico with her. Okay, she goes to the college clinic and she's talking to the PA about the fact that she has these really bad abdominal cramps. She has a fever, she's very nauseous, and she has bloody diarrhea. But she also has mucus mixed into her stool as well. Okay, so Brianna is presenting all the common symptoms of dysentery. Yes, that same dysentery that you can die on in the Oregon Trail game with. Okay. Now, the nurse practitioner that's seeing her collects some stool samples and sends them to the lab for analysis. Now, each of the stool samples are going to be used to make bacterial cultures like we've talked about in the course so far, but all these cultures are going to come back negative for any bacterial pathogens. So it's not bacteria that is causing the problem. So if it's not bacteria, it's usually something that's eukaryotic. So the next step is to start to do microscopy, because eukaryotic organisms are a lot easier to see underneath the microscope. Okay, a sample of Brianna's stool is going to be used to make a microscope slide, and they're going to perform a trichrome stain on it. Okay, this is going to reveal that Brianna has an infection of amoebas. Now, these amoebas are in a very specific state. They are what's called the active feeding or trophozoite stage. Now, amoebas, they like to get into your colon and they like to feed on the red blood cells there. Okay, in fact, if we look here at this amoeba, we can literally see its nucleus, right? It's eukaryotic conformation. And you can see these red dots. These red dots are the red blood cells that they have eaten. Okay, now the appearance of amoebas is characteristic of Entamoeba histolytica, okay, which is a parasitic amoeba that causes bloody diarrhea or something called intestinal amoebiasis. Okay, so once they have this diagnosis, Brianna is given a treatment called paramomycin, Okay, this is going to be a treatment that stops the protein synthesis. It has some pretty terrible side effects, like every eukaryotic treatment that we've talked about. So she's on this drug for 7 days, and her symptoms go away. Okay, so Brianna has the parasite Entamoeba histolytica. Well, the number one question is how did Brianna pick up this parasite while she was on vacation? Well, Brianna broke the number one rule of visiting Mexico in certain places. She drank the water. Okay, so she was on the beach enjoying drinks with friends, and one of her drinks contained contaminated water. Okay, getting this amoeba is a classic case of the fecal oral route transmission. Okay, so she drank contaminated water with feces in it. Okay, so when she drank the contaminated water, she ingested what are called cysts. Okay, so these cysts are going to start working their way through Brianna's digestive tract and to finally get to the colon. Okay, but as they are making their way through the intestinal tract to the colon, they're going to start to leave the cyst, okay, in a process called excitation. Okay, when these cells excite, they're going to start to develop into the active feeding stage, so those trophozoite stage. Okay, the trophozoite stage can multiply. Okay, these trophozoites are going to live in the colon, snacking on your red blood cells down there. Okay, but we're also going to have some that are going to develop back into cysts. Okay, so this cyst is going to go dormant and it's going to travel through the colon and get out with the rest of the waste. Now, there are some cases where the trophozoites can actually leave the digestive tract and enter the bloodstream. And when it does this, it can invade things like the liver, the lungs, the brain, and cause a lot of severe tissue damage there. But one of the important things to remember about this is that with eukaryotic parasites, microscopy is going to be a huge part of the diagnosis process because we can see these really well underneath the microscope. Okay, we are going to quickly talk about two other diseases caused by amoeba. So the first one is one that's really popular with the media, and that's because it's kind of exciting, even though it's really uncommon, and that is primary amoebic meningoencephalitis, or a brain infection. We're talking about the brain-eating amoeba, Naegleria fowleri. Okay, you can pick up this brain-eating amoeba by swimming in contaminated water. So now, Naegleria fowleri will literally swim up your nasal passage to reach your brain when you swim in this contaminated water. Okay, so not common. It's going to cause things like motor problems, so you're going to be really shaky, you're not going to move very confidently, you are going to have some personality changes, you might have some insomnia. It all just depends on what's happening here. But one of the reasons why it's so popular in the media is because it's going to cause the host to have these zombie-like symptoms. Now, the second one is going to cause keratitis, which is a corneal infection, so an infection of the eye. So, acantha amoebas are going to contaminate your contact lens cleaner. Okay, so when you go to clean your contacts, if your cleaner is contaminated, you can actually put the acanthamoeba directly into your eye. So we can see here, we can literally see that it's starting to eat your eye. Okay, especially the keratin in your pupil and in the back of your eye. Okay, this is not real common, which is probably, if you haven't heard about it, it's probably why. Only about 2 in every 1,000 individuals that use a lens cleaner will have this happen to you. One of the important things to pull away from this is that if you wear contacts, you need to switch out your cleaner about every 6 months because you don't want it to get contaminated and and accidentally put a amoeba into your eye. Okay, so the next group we're going to talk about are the apicomplexans. This is actually a major group of the protist family that infect humans and other animals. What's really special about the apicomplexans is they have what is called the apical complex. This is a specialized structure that these parasites have that they can then use to attach and enter into the host cell. Okay, so just like the amoebas, they're intracellular parasites. So we have two diseases we're going to talk about that are caused by apicomplexans. We have malaria, which is caused by the Plasmodium species of protists, and they are transmitted by a mosquito vector. Then we're going to talk about toxoplasmosis, which is caused by the parasite Toxoplasma gondii. Okay, Toxoplasma gondii is a foodborne parasite. Now, with malaria, we don't have to worry about this as much. Malaria is not found or is not endemic to the US, but toxoplasmosis is. So that's the one to worry about more between the two. Okay, so let's talk about malaria in more detail. So malaria is caused by a family of parasites called the Plasmodiums. Okay, we have different ones, and they're going to lead to different degrees of severity in the malaria that they are going to pass on. The most deadly is Plasmodium falciparum. Okay, and we're going to talk about this because even though malaria is not endemic to the US, it is still probably one of the most important and impactful parasitic diseases worldwide. So about 8.2 billion people live on the earth. Okay, 40% of where the world population lives is areas where we're going to find malaria. Okay, so in 2019, there was over 200 million new cases reported of malaria worldwide, and an estimated 400,000 deaths from it. The thing is, malaria is mostly going to affect young children. So out of those 400,000 deaths, about 250,000 of them were children underneath the age of 5. Now, 2019 has been a while. The most recent data we have is from 2023, where we have over 597 deaths worldwide from malaria. So we see an increase of it since 2019, and we see a rise in deaths in children underneath the age of 5. So we're talking about 375 to 400,000 deaths of children in that 597,000 deaths in 2023. Now, malaria is an ancient disease. It was first written about, if I'm correct, by the Romans. In fact, the Romans are the ones that named malaria. Okay, they use the term mal, which means bad, and area, which means air. So the Romans were the first ones to really start to associate malaria with swamps and swampy areas. Okay, now the Romans thought it was the bad air coming off the swamp. They didn't realize it was the mosquitoes that were biting them in the swamp that was actually transferring this parasite. Okay, now, as I said, it's transmitted by insect vector, by mosquitoes. Now, this Plasmodium falciparum is going to get into humans, and it's going to affect both the liver and the bloodstream. Okay, so let's talk about some symptoms if you're not familiar with malaria. So malaria is spread by blood meal, right? The mosquito lands on a human, it takes a blood meal, and it's going to transfer Plasmodium to the host. Now, once you get an infection and malaria starts, you are going to have a really high fever, but even though you are burning up to the touch, you are going to feel chilled and you're going to have these shaking chills. Okay, so it's going to present a little flu-like, right? It's also going to have symptoms of vomiting and diarrhea and tiredness. Because Plasmodium falciparum is going to start to affect your red blood cells, it's going to cause them to burst, so it's going to lead to anemia and low iron content in your body. And if it gets into your liver cells, it's going to start to cause damage to the liver, which can lead to jaundice. You're going to start to turn yellow. The first sign of this is going to show up in the whites of your eyes. Your whites of your eyes are going to turn yellow. And if not treated, hey, you're going to see some pretty severe symptoms. You're going to go into kidney failure. You can suffer from seizures. Um, you can have bouts of mental confusion, kind of like Alzheimer's. You're not going to know the people around you. You're not going to know where you are. You might not even know who you are, depending on the severity of the case. Most people who are untreated for malaria are going to slip into a coma, and a lot of them that go into coma are going to actually pass away from this disease. Now, as I said, malaria is not found in the US. Where we do find it the most is in Africa. In fact, malaria is one of the biggest killers of people in Africa behind AIDS and HIV. Okay, but we also see malaria a lot in Southeast Asia. Now, we did have malaria come to the US, and it wasn't endemic, it was brought over by people who were infected with it. And it got into our mosquito population here. Now, we don't see it much because back in the 1800s and in the early 1900s, there were a bunch of preventive measurements that went in that really kind of decreased your chance of getting malaria here. Okay, we drained swampy conditions. We dried out the land. We made sure there was no standing water. No standing water means the mosquitoes can't breed. If there's not as many mosquitoes, you can't pass it on. In the early about 1950s, there was these big campaigns that used insecticides to kill the mosquitoes off in different areas. So places like the South and Hawaii were hit the hardest because they're the ones with more swampy areas. And those preventative measures really kind of got rid of malaria here in the US. Now, Plasmodium, it doesn't matter which species of Plasmodium. It can be Plasmodium falciparum, which is the most dangerous. It can be Plasmodium vivax, which is one of the most common ones. It's not going to be as deadly as falciparum, but it's going to be actually the strain that we see in most individuals with malaria. Okay, so Plasmodium has an apical complex. Okay, so this is it in its merozoite form. Okay, so this apical complex is going to help it attach to the red blood cell. Now, red blood cells are really interesting because they don't have antigens, aka they don't have proteins on the outside, other than what's going to differentiate them as A, B, or O blood. And O blood has none, which is why it's called O blood, and AB blood. Okay, so it's going to use this apical complex to get itself into the red blood cell, and this is actually really dangerous. Okay, so these circles are this is a micrograph. These circles are actually the apical complex, so we can actually see it in the ring stage here in the micrograph. This is what these dark circles are. Okay, this apical complex is what makes them an intracellular pathogen. Okay, now it's really dangerous for us because once Plasmodium is in the red blood cell, it is effectively invisible to our immune system. Okay, it's hidden. Okay, so it's going to go through and it's going to reproduce and fill the red blood cell until the red blood cell lyses, and then it's free to then go and enter other red blood cells. Okay, so this is the life cycle of Plasmodium falciparum. Now, as I said, this is the deadliest malaria species. Okay, it's found mostly in Africa. This is where it's going to be the most, and this is where it's the most fatal. Right, malaria is one of the leading killers in Africa. We have another one called Plasmodium vivax or P. vivax. Okay, spelling is down here. That is more globally widespread, and it's going to cause severe disease, but it's less severe than falciparum. Okay, but it's also harder to get rid of because it has a lot more dormant stages when it enters the liver. Okay, so you can relapse and have malaria months later, even when you think you got over it. Now, as I said, so Plasmodium species are going to have 3 different life stages in this life cycle. they're going to have where they enter the human liver cell. Okay, so they're going to enter the liver cell, they're going to infect it, they're going to start to eat the red blood cells in the liver and the liver cells. They're going to form this giardia. Okay, the giardia is going to rupture, which is going to release Plasmodium into more cells and allow it to spread. Okay, from here it can actually enter the bloodstream. Okay, and this is the erythrocytic cycle. Okay, erythrocytes are blood cells. Okay, so it can then come into its trophozoite, its active feeding stage. Okay, that ring stage we saw in the micrograph before. Okay, it can mature into a mature trophozoite. Okay, it can then continue the cycle, forming the giardia, rupturing, and then infecting the bloodstream and going into more red blood cells and more red blood cells and more red blood cells. Okay, in the blood stage, it can actually enter into sexual reproduction. Sexual reproduction is going to make sure that this parasite has some genetic diversity, which is then going to lead to evolution. Okay, so it's going to produce gametes. Those gametes are going to go into the reproduction stage. Okay, so this is going to be what's found in the mosquito mostly. Okay, so we're going to make sporozoites and oocysts. Okay, now what's really interesting here is because it goes through sexual reproduction, it has a lot of genetic diversity, which means it goes through evolution. This evolution has actually been pretty bad because it's led this parasite to be resistant to a lot of the current treatments. In fact, billions of dollars have been spent to find treatments, to find vaccines, and to prevent malaria. Okay, so one of the original treatments for malaria was quinine. Now, the interesting thing about quinine is that it's an ingredient in the tonic part of the alcoholic drink gin and tonic. Okay, so people who sometimes drink gin and tonics are sometimes going to joke that they are preventing themselves from getting malaria. But the thing is, because of evolution in the parasite and genetic diversity, Plasmodium falciparum has actually become resistant to quinine, and it's no longer an effective way to treat it. We have spent billions of dollars developing treatments and vaccines. In fact, Bill Gates, the original owner of Microsoft, has invested billions of dollars himself. In fact, I think he did over 250 million in the last couple of years alone. So he's been spending a lot of money trying to help in getting rid of this disease. Now, as of a few years ago, most of what we had were ineffective treatments. Okay, sometimes the treatments worked, sometimes they didn't. In 2021, on October 6th, we actually saw the first malaria vaccine go into approval and go into effect. It was mostly developed for 5-year-olds or younger in Africa, and it's for one of the really, really deadly strains of malaria. In 2023, 2 years later, the WHO also approved a second malaria vaccine for use in countries that were endemic for malaria. Now, there was some other ones that have been in development, but I haven't heard much about those since those. But we do have 2 vaccines for this now that we didn't have before 2021. Now, most of these are going to be given to people in Africa, and they're going to be mostly administered to children, since children are the ones that die the most from this disease. Now, because it's so hard to treat, the easiest way is to prevent it. Okay, so we used to spray insecticides. So insecticides kills the mosquitoes, it keeps it off of you. There's huge campaigns in Hawaii and the South that literally would spray insecticides by planes, trying to cover large areas in order to get rid of the mosquito population. Now, we don't really have many malaria cases that are originate in the US anymore, and it's mostly because of those insecticide campaigns. Now, if you go to places that are endemic for malaria, a lot of people are going to sleep with mosquito netting, okay? Because the number one prevention way to keep from getting malaria is to prevent the mosquitoes from getting on you and biting you. Okay, so mosquito netting, bed nets, um, bug insect repellent, these are all going to be ways that you can prevent it. Now, there are some treatments called anti-malarials that you can take to prevent from catching malaria in these malaria regions, but pretty, they have some pretty terrible side effects because, yet again, we're treating something that's eukaryotic, and we are eukaryotes again. So anti-malarials can't be taken indefinitely, but you can always sleep with a mosquito net. Okay, let's move away from malaria for a bit and let's talk about toxoplasmosis. Okay, this is caused by the parasite Toxoplasma gondii. Okay, so Toxoplasma is a intracellular parasite. It's an apicomplexan, just like malaria, which means that it has the apical complex that's going to allow it to enter the different cells. Now, the thing about Toxoplasma and a lot of eukaryotic parasites is that they have complex and very interesting life cycles. And a lot of times, their life cycles are going to require specific hosts, okay, especially for their stage of sexual reproduction. Toxoplasma is no different. In fact, the animal that it prefers when it goes into sexual reproduction is cats. Okay, so Toxoplasma is going to enter into the cat host. It's going to produce its gametes. The male and female gametes are going to merge, and it's going to make this oocyte. Okay, this oocyte is actually a really super resistant structure that then gets shed by the cat. Now, the way it sheds it is it comes out in its feces. It comes out in the cat poop. Okay, now if you have a robust immune system, like most of the world, you are going to be asymptomatic even when you have this parasite. In fact, about 30% of the world's population is infected with Toxoplasma gondii. Okay, now why do we study and why do we learn about this? And that's because people like those who have AIDS, Toxoplasma gondii can lead to severe central nervous system disease. Okay, so it can affect those with low immune systems, but mostly it can affect pregnant women. So Toxoplasma gondii, if you are a host for it, even if you're asymptomatic, if you are female and you get pregnant, you can actually transfer the parasite to your unborn child through the placenta. Okay, now when Toxoplasma gondii goes through the placenta, the unborn child does not have a real great immune system, which means it can't fight the infection. And Toxoplasma gondii is going to try to enter the brain, where it's then going to cause developmental problems for the fetus. Okay, even in older individuals, it can also enter the brain and alter host behavior. Okay, you should never, never handle cat poop if you are pregnant. Okay, so once the cat sheds the oocyte, it's going to kind of contaminate the environment. Okay, it can be uptaken by mice. It can be picked up by pigs and sheep. It can actually enter the tissues of pigs and sheep, which then can enter humans when we eat them. Mice can pick it up from the environment, which is then going to keep it in the cat population as they kill and eat the mice. Cat poop can get into things like fertilizer, which then can contaminate vegetables. Cat poop gets into the water supply, which then contaminates the water. Okay, so this is a foodborne and waterborne transmitter. Okay, now what's really interesting is when Toxoplasma enters the brain of, say, like mice, they are going to kind of display zombie-like properties. They're going to slow down, they're going to have some mental confusion. These slower mice are actually really easy for the cats to catch, which is why cats are a great place for it to undergo its sexual reproduction cycle. Okay, there is actually some studies of humans who have infections of Toxoplasma gondii. Their behavior really changes, and there's been some connections that suggest a link between a Toxoplasma infection and some cases of schizophrenia. So it really does change the host. Um, it can also so not only can it get into the brain, it can get into the eyes and cause blindness. It can cause heart disease. It's not one that you want to pick up. Okay, so let's talk about our last subject of this lecture, and that is the extracellular parasites. So we're going to talk about trypanosomes and metamonads. Okay, so trypanosomes are specifically vector-borne parasites. They're transmitted by insect vectors. Okay, we talked about leishmania and how it's transmitted by sandflies. We have Trypanosome cruciae, which is going to cause Chagas disease. But the one that I want to talk about the most, and this is the one you'll see on your homework and on your test, is T. brucei, so Trypanosome brucei. So brucei is going to cause what is called African sleeping sickness. Now, African sleeping sickness is actually really important to know about because it is the second biggest killer in Africa behind AIDS. It's even worse than malaria, and malaria is up there. Okay, so African sleeping sickness is actually transmitted by the tsetse fly, so TSE, TSE. Okay, and it's done by blood meal. Okay, so we have our uninfected tsetse fly. It's going to land on this cow. Now, what's important to note about sleeping sickness is it doesn't only affect humans, it infects livestock as well. Okay, so the tsetse fly is going to take a blood meal from the infected cow. Okay, the trypanosome is going to go from what is called its stumpy form to its procyclic form. And once it's in its procyclic form, it can start to multiply. So it's going to multiply a lot in the tsetse fly's gut. Okay, it is then going to convert forms from procyclic to the epimastogote. Okay, and it's going to just continue to multiply. But instead, in the gut, it's going to multiply in the salivary gland of the tsetse fly. Okay, so when the tsetse fly lands on another mammal, so either livestock or a human, it's then going to take a blood meal, and this is going to be what transfers this trypanosome to its new host. Okay, it's going to be in what's called the slender form. Okay, this is what the slender form looks like. So it's this long, skinny parasite with this flagella. Okay, so it's then going to infect the host. Okay, where it's then going to multiply in the bloodstream. Okay, it's going to proliferate or multiply, and it's going to start to cause a lot of cell damage. And specifically, it's going to cause high fever that's then going to damage the central nervous system. Okay, this damage to the central nervous system is going to cause the host to experience increased drowsiness. This is the sleeping sickness part of the disease. And drowsiness is going to increase until the host slips into a coma. Okay, if untreated, this trypanosome infection can lead to death. Now, what's really interesting about trypanosomes, especially Trypanosome brucei, is they have something called high antigenic variation. Okay, so at the surface of this parasite, the cell membrane contains lots of different types of proteins, and these many different types of surface proteins on the cell membrane is going to make it so that our immune system has a hard time recognizing it as something that's a source of infection. Okay, so definitely know T. brucei and know about the tsetse fly. In fact, I'm pretty sure this is a question on your homework. Okay, so the last one we're going to talk about are the metamonads. So this is actually one that we can be concerned with because we have a metamonad in a lot of our water here. If you go and take a sample from the Snake River down in the river bottoms, there's a good chance that you're going to get the parasite Giardia lamblia in your water sample. Okay, so metamonads are flagellated parasites. They have lots of flagella, which means they're good swimmers. Okay, so Giardia lamblia is going to be transferred via the fecal oral route, so it's found in contaminated water. Okay, Giardia is also referred to as beaver fever because not only can it infect humans, but it also infects animals, including beavers, and hence the name beaver fever. Okay, it's going to cause gastrointestinal disease. So if you drink contaminated water and get this parasite, you're going to experience symptoms like cramps, lots of gas and bloating, diarrhea, and tiredness. Okay, there are some people that get Giardia, the Giardia parasite, and are asymptomatic, but they're few and far between. If you like to go hiking and drink water out of rivers, make sure that you are taking a water bottle with a filter. 0.2 micrometers or smaller in the filter will actually prevent Giardia from getting into your water bottle, because this is one that you don't want to have. So that is the end of the lecture. Okay, we're just going to talk about eukaryotic parasites and treating them as a whole, because it's completely more difficult than treating bacterial infections. Part of this is because eukaryotic parasites are eukaryotes just like we are. So they have similar cell structures and there's not a lot of differences. So a lot of the treatments that we use to target these parasites are also going to target our own cells. Okay, which means that we have lots and lots of bad side effects. Okay, and sometimes the treatment is almost as bad as the infection with some of these drugs. Okay, and then they have these really complex life cycles that makes preventing these diseases tough. And getting rid of these eukaryotic parasites, a lot of them can go dormant and hide, which means that even though you take the treatment and get through it, you might not have gotten rid of the parasite. As we saw with falciparum, it has a lot of genetic diversity and it's evolving, and it's actually evolving to become resistant to a lot of our treatments. So complex life cycles are a good thing for us. Okay, so that is 11.3, and we'll see you in the next lecture for 11.5.